Over the course of their distinguished careers, this year's Shaw Laureates, Swee, Thein, and Stuart Orkin, made seminal discoveries that led to the development of a therapy to treat sickle cell disease and beta thalassemia, debilitating blood disorders that affect over 20 million people worldwide. Blood cell sickling occurs due to low oxygen levels that are caused by abnormal hemoglobin. Hemoglobin is the protein in red blood cells that transports oxygen throughout our tissues. Important in the context of our laureates' work is that in humans, shortly after birth, a switch occurs from the fetal form of hemoglobin to the adult form. It is the adult form of hemoglobin that is defective in sickle cell and beta thalassemia patients. Swee Thein discovered that most variance in the severity of sickle cell and beta thalassemia disease is due to a single gene called BCL11A. Dr. Thein showed that the BCL11A protein is the major regulator of fetal hemoglobin production. Her discovery suggested that manipulating BCL11A could be a strategy to reactivate fetal hemoglobin production in adults and counteract sickle cell and beta thalassemia disorders. Stuart Orkin established that BCL11A turns off fetal hemoglobin production. Dr. Orkin identified a DNA sequence that, when altered using CRISPR genome editing in a mouse blood stem cells, lowered BCL11 production and turned fetal hemoglobin production back on, curing the disease. This work formed the strategy for human clinical trials. The trials yielded remarkable results: freedom from disease. Today, 2 sickle cell therapies are approved based on Swee Thein and Stuart Orkin's findings. One of them is the first ever approved therapy that uses CRISPR genome editing. Swee Thein and Stuart Orkin's work exemplifies how basic discovery, disease research, and translational medicine can drive development of transformative therapies that save lives.